Okay, this is the second half of the first section of the methods in the lumber biology lab. And in this section, we're going to be looking at some magnifications of but not microscopically. We're going to be looking at some photographs of pieces of wood at a couple of magnifications. So we're going to look at oak, spruce, yellow poplar, and palm. Why did I choose those four species? Oak is a ring porous angiosperm. Spruce is a gymnosperm. Yellow poplar is a diffuse porous angiosperm. Palm is a monocot angiosperm. That's why I chose those four species. So let's take a look at some of those. Okay, this is oak at probably 1x. This is how you'd see it with your naked eye. So what you can see, even with your naked eye, those early wood vessels in oak are enormous. Oak is one of the easiest wood species to identify because it's ring porous. It has these enormous early wood vessels, and it usually has really big files of ray cells. So with your naked eye, you can usually see the ray cells and the early wood vessels in oak. So that's oak basically with your naked eye. This is oak probably at about 10x magnification, and at this magnification, you can see both the early wood vessels as well as those late wood vessels. Okay. This is a spruce. So this is a piece of spruce wood, basically viewed with your naked eye. You can see early wood and late wood just by the color, but tracheids are really small, so we don't really see the individual tracheids at this magnification, at 1x magnification. But if we look at that spruce under about 10x magnification, we can see early wood tracheids, late wood tracheids, and files of ray cells. This is one growth ring here. All this fuzzy stuff is just places where the cuts across the face were not clear. This is that same cross section, but at about 20x. So here you can see better, the individual tracheids in the early wood. The tracheids in the late wood still sort of can't pick out individual tracheids and files of ray cells. This is yellow poplar. Okay, yellow poplar is an angiosperm. You can see growth rings and a little bit of gradation in the early wood and late wood, but not real apparent. Again, remember, this is a diffuse porous species, so those differences between early wood and late wood are not that apparent. Here's a 10x magnification of yellow poplar. This is not a great image because, again, the cut is sort of rough, but in here you can see, those vessel elements. These are vessel elements in there. This is a growth ring across in this direction. And then these are files of ray cells. So you can see the rays in this image. Really can't tell much difference between early wood and late wood, again, because it's a diffuse porous species. Here we're zooming in a little bit more on that yellow poplar, about 20x. Again, these are vessel elements in there. And this is palm. So this is a palm cross section, naked eye. What we're seeing, these dark spaces, these are all vascular bundles. We zoom in about 10x at that palm cross section. You can see the vascular bundles better. This is an old hunk of wood, so you can see the vessels really well, but the phloem is sort of smudged. It's not real obvious. Zoom in about 20x, and again, you can see the vessels and the vascular bundles here. The phloem is just sort of in this region. Okay, so take a look at those images that we provide for you, and then read the instructions in the lab report. So basically it says in your results and discussion, compare and contrast the xylem anatomy of the different species. So basically, I want you to produce figures from all of these photos that I give you, label them well, and then use those figures in your lab discussion to compare and contrast xylem anatomy in all of these different species that I've shown you. So use the concepts from lecture that I've shown you in terms of diffuse porous and ring porous angiosperms, monocot angiosperms, and gymnosperms, and how they compare and contrast, what cell types you see, and how cell types vary across the growth ring for those different species. I'm really looking for you to develop a story basically in your lab report that uses the images that are well labeled and formatted and refers to those images in your results and discussion to help the reader understand xylem anatomy in these contrasting species.